her master's thesis today. She has a 40-minute-ish talk to give to us, and then we'll take any questions from the audience and excuse those not on her committee to let her, you want to pull that close? So anyway, so I'm very proud of Nefertiti for getting to this point. It's been a bit of a slog, but you have persevered, and that has been awesome. So I'm gonna pretty much stop there, because if I do go any further, I'll get all mushy. So anyway, I'm very proud of you. Let's go ahead and see what you have for us.
Okay, so I was looking at, of course, the chloroplast twin arginine transport system, and specifically I was focusing on the composition of the pore. So, oops, there we go. And, okay, so CPTAT system is located, so in the chloroplast, it's located right on the thylakoid membrane. So I have this nice little cartoon of the chloroplast. There are many different transporters on the inner envelope, some on the thylakoid, but the CPTAT system is very unique. It transports fully folded proteins and uses the proton motive force, that electrical chemical gradient across the thylakoid membrane used during photosynthesis. So I'm really going to be focusing on that, but before we get into that, we're going to do a little bit of review and just go over some of the transport systems. So within the chloroplast, there are many places where proteins need to be transported. It could be the inner or the outer envelope, it could be the stroma, it could be the inner membrane space. But there's a unique situation. So the chloroplast actually descended from cyanobacteria, and it was engulfed by a eukaryote. So over time, the majority of its genome was transferred to the nucleus of the eukaryote. And the majority, about 95% of its proteins, are translated on the cytosol. So here I have this nice cartoon of some precursor polypeptides. So the question is, how do they get properly sorted to the chloroplast, whether it's the stroma, or maybe it's the lumen of the thylakoid. So right on the bottom of these polypeptides, we have this yellow, some yellow bars, we have some blue bars, and those are called transit peptides. So transit peptides are very important for the proper trafficking of these proteins. So you can think of them as a zip code or an area code. It directs them to the proper transporter, so they're sorted to the right compartment. So it's right on the N-terminus, and there's 3 basic regions. So on the N-terminus, there's an uncharged portion, about 10 residues. The central region is enriched in serine and threonine, but it lacks acidic residues. And at the very end, towards the C-terminus, we have a region enriched in arginine. So this would be a transit peptide, and then we have a bipartite transit peptide. So this means that one of these proteins, for instance, this one in the middle, has to go through maybe the inner and outer envelope, and then it has to go to the thylakoid. So there's 2 signals for it. Okay, so now we're just going to go over some of the transporters. So on the outer and inner envelope, we have TOC and TIC. So it's very interesting. So they're located on different membranes, but they work in tandem to transport unfolded proteins into the stroma. And then right here, it's kind of hard to see, but where my green light is, but there are these little scissors. That's an enzyme, and it cleaves off the transit peptide once it reaches the stroma. And if it's supposed to be trafficked to the stroma, then there won't be another signal, and it'll be properly sorted. But sometimes it will reveal a second signal, and it can be transported either through the TAT pathway, the SEC pathway, SRP, or the spontaneous insertion pathway to the thylakoid. So spontaneous insertion and the SRP pathway, they insert membrane proteins right into the thylakoid membrane, and SEC pathway and the TAT pathway, which we'll talk about later, they transport proteins right into the lumen. So we'll go over a couple of these before we get to the CPTAT system. Okay, so we're going to look at the TOC system. So this stands for translocon at the outer chloroplast envelope membrane. We know some information about the mechanisms, the proteins that are involved. So TOC is composed of, has 2 receptor proteins, TOC34 and TOC159 in the maroon and the gray. And then TOC75 is a beta barrel protein that's responsible for transporting it to the inner membrane space. So the receptor complex, it's about 800 kilodaltons, and all three proteins exist in a ratio of either 4 to 1 or 3 to 3 to 1. It uses GTP hydrolysis to thread the protein into the IMS. And for the basic mechanism, at first the receptors, they interact with the pre-protein, the transit peptide, and I have my cartoon these little G domains. So once it interacts with it, it changes a GDP nucleotide for a GTP, which undergoes nucleotide hydrolysis. And this leads to a conformational change, and it'll transfer the pre-protein to the beta barrel protein, where then it's threaded through. Then at the bottom, I do have TIC22. It's part of the TIC translocase, but it kind of guides the protein to the TIC complex. Okay, so then we come to TIC, translocate the inner chloroplast envelope membrane. We don't know a lot about the mechanism, but we've identified some of the proteins involved, TIC20, 21, 56, and so forth. However, in a recent study, they were able to identify, I think, about 4 different TIC components, and it was migrating or was pulled down with the precursor. So they think this, at the bottom, a 1 megadalton complex, that makes up those are the core proteins of TIC. So I'm not sure about the mechanism, but we think these are the main players. So 56, 20, 214, and 100. And TIC20 is supposed to be was predicted to make up the channel. TIC40 and 110, they might have a scaffolding role or a secondary role. And the last thing about TIC is that it uses ATP hydrolysis instead of GTP hydrolysis. So there might be an ATPase in the stroma that's helping to thread the protein through.
Technology. Yeah.
Technology?
Just use the, oh, it froze.
But if you can get one that's new between mine and one thing, no, I don't know if I agree with this argument.
There we go.
It went? Okay.
Yep.
Okay. So now we're going to go to the thylakoid. So after the transit peptide is cleaved, if another one is revealed, these blue bars, for instance, then it could go, then it's supposed to be sorted to the thylakoid. So we're going to talk about spontaneous insertion first. So we're talking about membrane proteins being inserted into the thylakoid membrane. So what's interesting about this is that there's no energy requirement, there's no nucleotide hydrolysis. There's no insertion machinery no translocation apparatus. Some of the substrates are very relevant though. So we have some from photosystem 1. We have one component from the SEC pathway, which we'll talk about a little bit later. And then from the twin arginine, from the CP-TAT system, HCF106 and THOT4 are spontaneously inserted, and possibly CP-TAT-C. Okay, so the next one is the general secretory pathway, or the SEC pathway. So SEC pathway, it transports proteins into the lumen. So we're not inserting them into the thylakoid membrane. There's 3 basic proteins, CP-Sec-Y, Sec-E, and Sec-A. Sec-Y and E make up the translocase, and Sec-A has multiple roles. So it recognizes the transit peptide of the pre-protein. It also guides the pre-protein to the channel. And then it's an ATPase. So it hydrolyzes ATP in order to thread it through into the lumen. So the mechanism is pretty simple. Sec-A, it recognizes the pre-protein. Like I said, it guides it to the channel. And with ATP hydrolysis, I think there's conformational changes so that the protein is threaded into the lumen. Okay, so finally we come to the CP-TAT system. So it gets its name from the characteristic twin arginine motif on the transit peptide. So I didn't put that there, but Yeah, so right over here, it would have the twin arginine motif, and if it was changed to lysines, for instance, even though arginine and lysine, they're kind of similar, but if it was two lysines, the receptor complex would not recognize the precursor. So CPTAT is made up of 3 proteins. CPTAT-C is in blue, it spans the membrane 6 times, and HCF106 is in green. And as you can see, both of those are together in a separate complex, the receptor complex. And that migrates at around 700 kilodaltons. Then in panel A, you see off to the side, we have Phi4 in pink, and they exist as separate oligomers. So that's the beginning of the, CPTAT, it works in a cycle. So this is the beginning of the cycle. We go to panel B, the precursor is recognized by the receptor complex. It recognizes that twin arginine motif. And then in the presence of the proton motive force, that energy gradient across the thylakoid membrane, Phi4 will oligomerize within the receptor complex, and that forms the active translocase. CPTAT is different from the other transporters because it doesn't use any nucleotide hydrolysis. So that's one thing that makes it unique. The other thing is that it transports fully folded proteins, and we'll talk about that a little later. So after the active translocase is formed, the precursor is transported into the lumen, and then in panel D I have this little Pac-Man that's the peptidase that cleaves off the transit peptide. And after the protein is transported, the system basically resets itself. So Phi4 disassembles and it goes back to being separate oligomers, and then you have the receptor complex that's ready to receive another preprotein. Okay, that looks good. So, like I said, CPTAT transports fully folded proteins. So these proteins could be from anywhere from 2 kilodaltons to 100 kilodaltons, 20 angstroms in length to 70 angstroms. And so one reason why they're folded might be because they rapidly fold after they're translated, or maybe there's not enough folding chaperones around. But one question that we have is, how does it transport these really large proteins without damaging or damaging the integrity of the membrane and causing ion leakage, or just dissipating the proton motive force? So we're just going to take a closer look at the pore. So Phi4 is the one in pink. Phi4 is predicted to make up the pore. There's a couple of reasons for that, but let's look at the structure of Phi4. So it spans the membrane 1 time. We have a short transmembrane domain near the N-terminus. There's a hinge region that connects it to this amphipathic helix that kind of tilts into the membrane, and at the end we have this largely unstructured C-tail. So one reason it's predicted to make up the pore is that it exists in molar excess over CPTAT-C and HCF106. They form large oligomers, and like in the other model you saw when they oligomerize, they kind of looks like they could form a pore or form a channel. It interacts with the precursor late during transport, and it's also required for the transport step. We also know that it binds directly to the mature domain of the precursor, but we don't know for sure if Phi4 is the only component of the pore. So if we look at HCF106, it has a very similar structure to Phi4. Both of them, they're homologous structures, as a matter of fact. It has the short TMD, a hinge region, an APH region, and it has a larger C-tail, but they basically have the same structure. So we also know that HCF106, it also forms oligomers. It interacts with Phi4 in the absence of the precursor. Like I said, it's homologous to Phi4, and it also binds to the precursor. So what I was looking at is which regions of HCF106 interact with the precursor. Is most of the interaction the C-tail, the APH, is it in the N-terminus? So I used cysteine cross-linking to create an interaction map with HCF106 and one of the substrates of CPTAT. So right here on this side, this is oxygen evolving complex. It's involved in splitting water during photosynthesis. It's a CPTAT substrate. So I'm looking at interactions between oxygen evolving complex and HCF106. So with cysteine cross-linking, I'll make different cysteine substitutions on different parts of this 4-bundle helix. And then and I'll do the same thing with HCF 106. So where these arrows are, there's about 20 different cysteine substitutions that were made. So under transport conditions, if they're in close proximity to each other, a disulfide bond will be created, and I'll know, okay, they're interacting with each other. So I'm hoping to find out which region of HCF 106 is mostly interacting with the precursor. Okay, so this is just a rough summary of the methods. So at the top I have oxygen evolving complex and HCF 106, and you can see both of them have a cysteine substitution, but only one of them is radio labeled, so I can look at it on SDS page. So HCF 106 is the one that's radio labeled with tritium. And then on the second step, I'll take a radio labeled HCF 106 and I'll integrate it with thylakoid. So then I'll do a transport assay. So with radio labeled HCF 106, I'll add my oxygen evolving complex. And then during transport, I'm hoping to get a cross-linked product if they're in close proximity to each other. And then on oxygen evolving complex, I have a histidine tag, and I use nickel affinity column to pull that down, isolate it, and look at it on SDS page. But
Yeah, I don't think I'll go.
on SDS page, I'll only see HCF 106. That's the only thing that's radio labeled. So I would hope to see something like this. In this first section, I would hope to see the cross-linked product that I would get from the nickel column by pulling down the precursor. And then to make sure that I have HCF 106 in my sample, I would take whatever was washed off the column to make sure it's still present. Then I also have the translation product to make sure that HCF 106 was translated. Okay, so with this figure, I'm looking at oxygen evolving complex, and the substitution is on D68. So I'm looking at, oh, okay, I see I made a typo. I said T115, that should be D68. But it interacts strongly with the C proximal APH and the C terminus of HCF 106. So in this lane, I have a positive control where I know that this variant of oxygen evolving complex interacts with this variant of HCF 106. And over here, this is D68 interacting with the N-terminus of HCF 106, and this is more the C-terminal APH region. So I only got a cross-linking product for S53. But from looking at previous studies, I know that the overall trend is that it's strongly interacting with the C proximal APH and the C terminus of HCF 106. Okay, so this one is with T115, that variant of oxygen evolving complex. And so I found that it only weakly interacts with two variants of HCF 106, S58C and I64C. So that's at the very end of the APH region near the C terminus. And so again, on this side, all of these columns, I have my positive control with another variant of oxygen evolving complex and one variant of HCF 106. So the only cross-linking I got was at the very end of the APH, and it's very faint. Okay, so just as a summary, this one is D68. This is the one that was interacting with the C proximal APH and the C tail of HCF 106. And T115 over here, that only interacted with two residues in the C proximal APH of HCF 106. Okay, here I have a nice summary of the interactions of HCF 106. So closer, like I said, closer to the at the end of the APH region in the C tail, there's stronger interaction. But in the TMD, there's no interaction. It's a very hydrophobic region, so that makes sense. And then in the N-terminus, there's some weak interactions. So for my conclusion, I was thinking, so I was really looking into whether HCF 106 plays a part in the pore, forming the pore. I think it might be involved in stabilizing the precursor versus actually helping it to transport into the lumen. So in this picture, I'm comparing an interaction map of HCF 106 and Phi4 from a previous study. So with Pha4, there are some green circles near the N-terminus, so there's strong interaction. Some places in the N-terminus, a little bit in the TMD, and also the APH region. We compare that to HCF106, there's only strong interactions in the APH region and some in the C-tail. So I think that the precursor is interacting with Pha4 throughout transport when it's bound here, and then when it goes across the membrane, it interacts in the N-terminus. But since it only interacts in the APH region of HCF106, I'm thinking that maybe HCF106 is just stabilizing it, and then when it transports, it's mostly interacting with Pha4. Okay, so that was the first part of my research. As I was leaving, I started a different project. I was looking at whether, so before I was just looking at cross-linking between HCF106 and the precursor, now I'm adding another element. So when oxygen evolving complex is in complex with HCF106, can it be transported? So while it's cross-linked, can it be transported? So I was thinking if it was still in contact with HCF106, maybe in the N-terminus, even after it's transported, maybe it has a role in stabilizing it while it's being processed in the stroma. Might be something like that, but I was curious about it. So in order to investigate this, I'm using thermolysin. So I basically have the same procedure from the previous study, from the previous part, but when it's cross-linked, if I add thermolysin, it's going to degrade anything that is exposed to the stroma. So if it's not transported and it's exposed to the stroma, the precursor will be degraded. But if it's membrane protected, I know it's been transported and it won't be degraded. So that's what I'm looking for. So like I said, the methods are pretty much the same except I'm adding thermolysin. So, well, there is one other thing that I did change. So instead of radio-labeling HCF106, I radio-labeled oxygen evolving complex. So this one's hot, that one's cold, but the histag is still on oxygen evolving complex. So I'm still going to pull that down with a nickel affinity column. So I'll take my cysteine substituted HCF106 and integrate it into the thylakoid. I'll add the precursor. This time the precursor is hot. And after transport, when the crosslinks are formed, I'll add thermolysin. So if it wasn't transported, if it can't be transported while it's cross-linked, then thermolysin will degrade it. But let's say there was a cross-link, but it was transported, then thermolysin, it won't degrade it. So I was hoping to get a cross-link product and then to pull down the precursor using a nickel affinity column. And then I would hope to get something like this. So everything you see here would be just oxygen evolving complex. You wouldn't see HCF106. Then I would have two columns. So for each variant of HCF106, after transport, I would split the sample into one. I would add thermolysin so I can compare the two. So if let's say that if I added thermolysin and this band disappears, this means that this wasn't transported, but this was, for instance. Then this would be my positive control. So I was hoping to see cross-link product that was transported. Okay. So with this figure, I was looking at T115, and then for my positive control, I had K99C. So it's kind of hard to tell on this screen. So HCF106 is at the top, and I'm looking at the N-terminus and oxygen evolving complex. Here, it's a very faint band, but there is some precursor here, and on the bottom, that's the mature domain where the transit peptide has been cleaved. And then I add thermolysin to this column, and the precursor disappears. So this would be transported. It's the same thing with I64. It's very faint. It's really hard to see it, but it's actually there. When I add thermolysin, the protein is degraded. And then, of course, this is my positive control. So I thought that I had cross-link product that did transport, but when I look closely at it, interactions between T115 and G6, there was no interaction for that when I did my interaction studies. So I said, oh, this must be uncross-linked precursor. So I have to change my methods. So the problem was, I had, the precursor was radio labeled, but it also had the his tag on it. What I should have done was maybe put a flag tag on HCF 106 and had this radio label. So at the end, I couldn't really distinguish between precursor that was uncross-linked versus precursor that was cross-linked. Because I'm pulling down precursor and both of them are radio labels, and that's all I'm seeing on the gel. So I can't tell whether or not it's cross-linked to HCF 106. So in the future, well, some revisions I would make, like I said, I would put a flag tag on HCF 106, and then whatever I pulled off of the column, it's going to have HCF 106, but I'll only see the precursor because that'll be radio labeled. Something else that I could have done if I had more time, I could have had the same samples, but I could remove B and E from the sample buffer. So the disulfide bonds, it wouldn't be reduced. So if I had any cross-linked product, it would migrate at a higher weight versus precursor that was uncross-linked. So I really could have done that, but. So coming to the conclusion, just looking at the overall model for CPTAT, and just how the precursor is transported without damaging the integrity of the membrane. So I know in previous studies, HCF 106 and Tha4 undergo conformational changes when they bind to the substrate. So when HCF 106, and this is also in bacteria too with TAT A and TAT B. So when HCF 106 binds the substrate, the APH becomes more parallel with the membrane, and that pulls the T and D towards the stroma, towards the stromal side. And the same thing happens with Tha4. First it's tilted, when it interacts with the protein, APH becomes more parallel and it pulls the T and D towards the stroma. So I think this causes some hydrophobic mismatch where the interactions between the lipids and the proteins, well, the lipids are trying to, I guess, match the thickness of the T and D regions. So maybe it causes the membrane to thin so much that it creates a transient rupture, just a small rupture, so that the precursor can go right through. So I think that that's probably what's happening instead of just having an aqueous pore. Maybe Tha4 and HCF 106, they just work together to destabilize the membrane so the precursor can go through. So that concludes my presentation. Just want to thank everybody in my advising committee, Dr. Dabney Smith, Dr. Hagerman, Dr. Lorgan, CNSB program, Department of Energy. Everybody in my lab was always super helpful when I was here, just teaching me things over and over again because I was a slow learner. And then when I first got here, my first year, I was working on a different project. Everybody from Dr. Lorgan's lab, they were also like really, really nice to me too. So thank you. Do you have any questions?
Very good. So I'll open it up to the floor for questions. Do you have any questions?
I do not. I'll say all to you.
I have a question. Can you go back to the very first diagram you showed of the SDS page?
This one?
That one.
That one, yeah. I'm confused right now about why the protein with the cross-linked two proteins isn't bigger than the unbound or the translation product.
Oh, it's because with the B and E, it breaks the disulfide bond, so we're just seeing HCF 106.
So it's the treatment when you put the protein on the SDS page, you use denaturation buffer and you break down those disulfide bonds.
Yeah, for the thermolysin experiment, if I hadn't put B and E in, that would have saw if I had cross-link product or not, it would have been a higher
I
beta.
get it.
Any other questions for now? Okay. All right, then I'm gonna